of all, I put my sincere thanks to our management of. Anbu sir, can you hear Anbu sir?
Yes, sir.
Yes, sir. Yes, sir.
First of all, I put my sincere thanks to our management of Kalasalingam Group of Institution for accepting wholeheartedly to conduct this 54th International Virtual Learning Series with the blessing of Almighty, our founder, Kalvi Vallal Devatru, the Kalasalingam, and with the support of our chairman, Ilay Vallal, Dr. S. Sridharan, sir, for the 54th International Virtual Learning Series. For welcome all on the behalf of HCP for this 4th International Virtual Learning Series. We are very proud to have an eminent researcher, scientist, veteran, and honorable resource person.
Thank you, sir.
Dr. Nagendran Tharmalingam, researcher associate, Mylonakis lab, infective disease division, Island Hospital and Alpert Medical School of Brown University, USA, to grace this occasion. It is a great pleasure for all of us that he could find time to be with us in spite of his busy schedule. I take this opportunity to convey my sincere thanks to you and your esteemed university for accepting our request despite of your busy schedule, for being a valuable part of this session. Welcome, sir. Welcome, Dr. Nagendran, sir.
Thank you, sir. Thank you very much.
Welcome, sir. Your prestigious presence, great motivation, inspiration for entire student community, sir. Once again, I welcome you, sir. Next, I welcome all the professor, faculty members, and my dear enthusiastic students for today's session. My dear students, this session hope will enrich your knowledge and update you about the drug repurposing to combat infectious diseases. This is the first and foremost needy for your career. All the very best, my dear students. Once again, I welcome you all. Thank you. Thank you, Dr. Nagendran, sir. Thank you, sir. Please,
You're
you can
most
proceed.
welcome, sir. You're most welcome, sir.
Thank you. Thank you.
Okay. Good morning. Very good morning, one and all. Sir, Anbu sir, can you see my screen?
Yes, sir.
Okay. So I just want to make sure everything is all right. Okay. Good morning, respected principal and professors, fellow colleagues, and all the students. It's my great honor to be a part of this session, and I'm really happy to present the data and then the research, the sources, what I am having in my, the acquired during the tenure here in this country. So myself, as principal mentioned, myself, Dr. Nagendran Tharmalingam. Basically, we both sharing a similar background. Obviously, I'm a paramedical staff. Initially, I started my career as a medical lab technician. I just finished my 1 year of certified medical laboratory course in Madurai Alpert College of Pharmacy, and then I did my bachelor's and master's in general microbiology in Providence University, where I acquired and I got a lot of knowledge, and the thirst the thirst towards research where it emerged at Providence University. We have a lot of very fantastic professors over there, and that seed the research over there. And then I went my to South Korea to do my PhD, and then I'm working here as a senior postdoc in infectious disease division. So the topic we have today is drug repurposing to combat infectious disease. Basically, I'm a microbiologist, and the thought from the beginning, I mean from the childhood, to want to find a new medicine to whatever it may be, but I choose microbiology as my career. So I'm working here, and fortunately I'm working here. Let's move on. So as per the definition we have, hold on a moment, please. I'm sorry. Yeah, we have a lots and lots of definitions towards drug repurposing and profiling and discovery. There is lots and lots of directions towards how we make this drug into a multi-purpose thing. So based on that, we have different aspects and different perspectives towards drug and how we can do. But what I do, like I'm focusing on a drug repurposing, which is identifying a drug, how it can be used in another purpose, and then how we can develop the identified drug and then make it toward to commercialization, how to commercialize the drug for the new users. So the drug, what it may be, orphan drug, abandoned drug. So abandoned drug is nothing but the drug which is in the old use, or people may forget the drug, or it may be in a poor use or poor tissue penetration, but it developed for the good purpose. However, the purpose which is not satisfying in a manner, so we totally forget. So that we call drug repurposing. We took, based on the whole screen, based on the whole drugs available in the screening, we did the assays, which is called high throughput screen assay. So with that having, like, if we say drug repurposing, that should be a like a approved drug or a FDA approved drug. So we have like actually based on screening like 1,950,000 something compounds, and then we did that assay, and then based on that high throughput screen assay, we got the hits. Based on the hits, then we scrutinized, we shortlisted, and then we took that drug, and then that can be developed in a good manner that it can be applied to a public. So that's a whole story about the drug repurposing study here. So as you all heard of Tufts University in United States, which is very near to me, like a mile, 1 hour drive from here in Boston. So they are very good medical school and drug developing center over there. They found that to develop a drug which is cost 2.1 billion, that means which is thousands of crores in INR, that cost the drug which came from what we call like bench side to bedside. That means a drug what we have identified and developed, that is in a bench side, and after the trials and clinical trials and then approval, then it will go to the it will prescribe to the patients, that is called bedside. So from bedside to bedside, it called like 2.1 billion. Can you believe this? So this is a fantastic study that conducted by the researchers in Tufts University. And then, as you can see here, over the decades, the drug cost is enormously increased, and then the manpower, the chemicals, and then the facilities, instrumentations, everything has increase in the prices. That results in a very high price of a single drug which costs 2.1 billion. So that's how the FDA, they suggested that, okay, why don't we repurpose the drug? So according to the FDA, as we all know that Food and Drug Administration, one of the clinical drug approved organization in this country, in the United States. So why they want to repurpose the drug? So first of all, the success rate, and then the difference between the drug repurposing and the de novo, which is a new drug developing strategy. The success rate is, as you all see here in the screen, that 1/10 of success rate in the drug repurposing, and And no wonder, no question, or no discussion, these are already approved drugs, so the safety is well known. When compared to the new drug development, the safety is unknown because the toxicity, tissue penetration, and PKPD, and then the plasma availability, and the clearance. We don't know anything about that new drug development. However, what we have is the drug repurposing. That new drug, that drug available already in the market. We know all the data. So according to that safety, obviously yes, we can trust this drug can be go to the next level. And and you all know that you guys all know that to develop a drug, it costs like much money, and then the time is also it's a time consuming process, and antibiotic that need at least 15 to 20 years that need to from the bedside to bedside. So based on all the factors, and in addition to the cost wise, and as you can see, this is very less, the cost level is very less, because a lot of studies we can cut short the lot of techniques and cut short the manpower to skip the studies, and then we can go to the next level. So based on that, yes, the drug repurposing, it's one of the fantastic strategy, and now that is the hot topic. And as you all know, a lot of researchers and the professors, and even the president of this country, he pronounced the name like chloroquine, hydroxychloroquine, and why this drug, and then the topic, why it is so emerged. It's because of this COVID, SARS-CoV-19 drug finding. So instead of finding a lot of drugs, the researchers, they come up with a solution. Yes, we have drug repurposing, and then based on that, our NIH, National Institute of Health in the United Nations, they are pouring a lot of money to the research right now. So as you can see, the clinicaltrials.gov, that in the website where you can go and see what are all the drugs which is in the clinical trial stage. That means whether it is a phase 1 or 2 or 3 or 4, and how it is, and what is the documents they have submitted, all you can see. So based on the data available right now, the drug repurposing thing that you can see here, that chloroquine, which is all you know, that the malarial drug, that drug which can be acting as a antiviral drug, that it goes to the infected cell, and then the pH of the pH of the cells, it can play with the pH of the medium, that results in the viral particle release. It disturbs the release, so that's why the virus couldn't be mature. So this is what the hypothesis mechanism behind that. And then in addition to the chloroquine, there is a lot of the drugs they are proposing for the repurposing. However, for the clinical trials, as you all can see, the Montelukast, the drug which they can prescribing for asthma, and then chlorpromazine that they are prescribing for the psychotic disorders. In addition to that, what is my cup of tea is like doxycycline. So I'm a bacteriologist. So doxycycline is a broad spectrum tetracycline antibiotic. So the research study that proposes that doxycycline that can stop the cytokine storm. And as you hear the term like cytokine storm, the virus infection, the SARS-CoV-19 virus, once after the infection, as the human system develops the hyperimmune system that results in cytokine storm. So they propose that doxycycline can stop the cytokine storm. So these are the topic. These are the drug they at present in clinical trials. Believe it, people, that getting into the clinical trial is not that much easy, but still they are doing this. That means that they have potential results, or who knows. Let's see the results. That's a much awaited thing right now. And then, repurposing drug to combat the bacterial infection. And before going there, the topic how we choose our lab. My boss here, he worked in a Harvard Medical School previously, like 6 years back. He was basically a fungal scientist, and then antimicrobial fungal scientist, antifungal finding scientist. Then he was working with the project that using a C. elegans, one of the infection model, and using that one as to screen the antimicrobial hits. So based on the hits, they find that one of the drugs, salicylate, group of drug called niclosamide and oxyclonoside. So those drugs, they can actively inhibit the Staphylococcus aureus. And then what we took that antibiotic and we coated the drug with in the urinary catheter to inhibit the biofilm formation. And yes, we got the publication. Then it was published in the Biomedical Materials, one of the good journal. They accepted our publication that yes, it inhibit the bacterial biofilm formation in the catheter. And also the other drug, ornithine. So ornithine is one of the orphan drugs. So orphan drug is probably you may have heard this terminology, orphan drug, that if you say for an antibiotic, that's a broad disease, and if you say cancer, it's a broad disease. If you say for psychotic, it's a broad disease. But very peculiar, unique diseases. If the drug that can treat only that unique, particular, very rare disease, that called orphan drug. So the ornithine, one of the anti-inflammatory drug which they can use for arthritis, that we found as one of the hit to inhibit the bacterial infection. That means it inhibit the MRSA infection, Staphylococcus aureus infection. So when we published that, and then that ornithine that also inhibit the fungal pathogens. And here is the one of the very interesting story here. So Fancicella tularensis, you may all hear about anthrax story. So anthrax is a bacillus caused by Bacillus anthracis. So that bacteria, which is a spore former and using as a biowar weapon. Believe it or not, this Fancicella tularensis is also one of the biowar pathogen. That what we got, based on the repurposing strategy, we found a drug called diflunisal. Diflunisal is also one of the anti-inflammatory drug that it can inhibit the Fancicella tularensis bacteria. That we found this through this high throughput screening, and we published it. As you can see my name, that I'm also one of the part of the project. And then yes, here is my baby. Repurposing the anti-helminthic drug niclosamide to combat Helicobacter pylori. Basically, I'm a H. pylori scientist person, but I'm dealing with this bacteria, the bacteria which is known for the gastric cancer inducing pathogen. So this drug, one of the anti-helminthic drug, which is comes under a salicylase group, that inhibit, 4 years back, I was doing that assays, and then I found it was really accidental story. And you know what, there is a story, as you all know, that the Eureka moment. So when the Archimedes was in the bathroom, and then he found it, and then he was running all over the road without any clothes, like the Eureka moment. So when I was doing this assay, so I found this drug in our lab, and then since H. pylori is my cup of tea, and then I was doing this assay, and then I did this assay, and then I found that after 3 days I found this. Oh my God, this drug is inhibiting the bacteria. So that will be a really good story, and that story is ending here. That ended here in Nature Scientific Reports. We published the paper, and then that story we're gonna discuss here. Basically, drug repurposing that can inhibit whatever. I mean, the purpose, inhibit whatever, or it can induce whatever. Here, my strategy. Basically, I'm a bacteriologist, a medical bacteriologist. The drug, it can inhibit the bacterial infections. I'm doing what I'm doing right now, and I'm going to present you right now. And then viral infections, as we discussed previously, where the drugs can inhibit the SARS-CoV-19 virus, and then fungal infections like the ornithine drug, it inhibits the fungal pathogen, and then we published that paper, and then it can inhibit the protozoal infection, probably, who knows, like antifungal can inhibit the protozoa, or antiviral inhibit the protozoa, who knows, but all we need to explore. So we have a lot of rooms. So here, I'm presenting the room here. The escape C pathogens. What is escape C pathogens? That is Enterococcus faecium, Staphylococcus aureus, Klebsiella, Acinetobacter, Pseudomonas, and Enterobacter classium difficile. We classifying escape C. And as you can see, the first letter E, Enterococcus, E for Enterococcus, S for Staphylococcus, are taking all the first letter, and they form a terminology escape C pathogen. You may hear of the terminology nosocomial infection, which is a hospital acquired infection. If when you're going for a surgery or procedure, whatever, like dialysis or any implants, due to the invasive surgeries, invasive procedure, there may be a high chance of getting infection from the implant or from the infection or from the surgery. So these are the 6 predominant bacteria, the first 6 predominant bacteria it may cause hospital-acquired pathogen. So these are all the bacteria that I'm focusing on. So it will be a really interesting story. And then the world need the drugs against these 6 first pathogens, and then the 7th one, the Clostridium difficile. So one of the diarrhea causing anaerobic pathogen, that also one of the included in the SCFC hospital acquired when there is a person who is under heavy antibiotic intake. That person have and then the person the preferred person who is more than 60 or 70 year age, like he is highly prone to this acquiring the Clostridium difficile disease. So this hospital acquired pathogen, the world need a drug against these 7 bacteria especially. And then the gastric pathogen, H. pylori. And as you know, no wonder I want to say that H. pylori is my cup of tea. Like I like this bacteria. That's my PhD job. I like those, so I'm working against that. And the Francisella tularensis and the Staphylococcus aureus, those are comes under intracellular pathogen. Basically, Staphylococcus aureus is the facultative anaerobic bacteria, and then the report says that it is also a intracellular pathogen. It can getting into the cell and then multiplies, and it can resist the host defense mechanism. And then there is a other room what we call antivirulence. So killing a bacteria is the strategy, and then there is one other strategy, which is a dismantle or disform the bacteria. That means bacteria producing a lot of virulence factors. That is the most, that is the culprit, that the human getting infected and then seeing the lot of symptoms of fever and even to the fatality, that because of all the virulence factors. So if we dismantle that virulence factor, yes, we can defend against the pathogen. So that's one other strategy. So this repurposing, that also working with that strategy. And then let's we focus more about H. pylori, and then seeing about what we have, and then why we against H. pylori. Why I am talking a lot about H. pylori? What is H. pylori? Helicobacter pylori. So World Health Organization, 2 years back, it publishes an article that these are the critical pathogen. The world, that all the researchers, we need to work towards these all pathogens because they are really highly emerging and dangerous the world gonna face. In other words, antibiotic resistance. As you can see, the priority number 1 critical, so Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacteriaceae. All these 3 pathogens considered critical, and as you can see here in the priority to high high priority level in the third content, H. pylori, clarithromycin resistance. So the World Health Organization itself says that this is the highly dangerous pathogen that we need to focus. Hence, I choose this one. Why don't we go for it and then do research against this? And the reason why the World Health Organization published this, why they need this, because of antibiotic resistance. And in 2050, it is an estimate that 10 million people, they may die in because of antibiotic resistance. And believe it or not, even in the 2050, we may have a cure to cancer. However, we may find difficulties to find a new antibiotic against this pathogen, and due to the antibiotic resistance emergence, we may face a lot of difficulties due to this antibiotic resistance. So that's why the World Health Organization, they publish this, and then they request us, like researchers like us, we need to work on it, and we are on a track. Okay, let's discuss more about H. pylori. So Helicobacter pylori, which is the, you all know that the basics of bacteria, like it's a gram-positive and gram-negative. So gram-negative are little more difficult and tricky to treat because of the complex nature and the complex structure, outer cell wall structure, outer membrane layer structure. The gram-negative pathogen, H. pylori, This is a helical shaped and micro aerophilic bacteria. That is, it is a stomach pathogen. It is a gastric pathogen. This bacteria survives in the acidic environment in our stomach. The bacteria, it is identified in 1982, and then let me tell you a very interesting story about this H. pylori. How the bacteria comes to the limelight. You know what? That will be a really interesting story. Believe me. Actually, in 1970s, Barry Marshall is a doctor, a physician in the Australia. If I'm not wrong, that the hospital name is Queen Victoria, I guess. But I'm sorry about the wrong information. I don't know. But let me tell this. He's a doctor. He's a physician. He was a resident student. I mean, at that time he comes under Robin Warren, and then they work together. He was seeing patients. He was seeing patients with gastritis, and then he developed a hypothesis why this gastritis may develop from a bacteria, and then everyone laugh at him. People laugh at him. So, however, he believed himself. He talked to Robin Warren. He's a senior doctor over there, and then they had a plan, and then they tried to isolate the bacteria from the samples. And basically, all the basic microbiology experiments that start with culturing a bacteria. As you all know, I'm sure that you guys all know about how to culture the bacteria. So they try to culture the bacteria. That is one of the very simple technique. However, they don't succeed. One Easter evening, so Easter Friday, Good Friday evening, before the Good Friday evening, Thursday, he got the samples and then he cultured in the plate and put it in the incubator, which requires, as I said, it's a micro aerophilic bacteria that requires only 5% of oxygen. It requires a 10% CO2. That is how our human body reacts to the gas. How it's mimicking our human system. So that's why they culture the bacteria and put it in the CO2 incubator. And then it was a Good Friday. It was a holiday, obviously, and Good Friday and Saturday and Sunday. And after 3 days, the Barry Marshall, he came to the lab on Monday. He was shocked. Believe it. What happened? You already guessed. That bacteria grows because H. pylori bacteria, which is a slow-growing bacteria. They didn't know that because even they don't know whether this disease is caused by a bacteria or not, because at the acidic condition, acidic medium, how this bacteria react, how the bacteria grows, they don't know anything. They have a blind thought that this gastritis or ulcer that can cause by bacteria, and they don't know. They did it in the Good Friday. The previous day of Good Friday evening. They culture the bacteria and then put it in the incubator, and after 4 days, they came and got the bacteria. What? That not even end of the story. They want to prove. He told this story to everyone. No one else believe. Then what he did? You can't even imagine. He drank the bacteria. He literally drank the bacteria. He cultured the bacteria and then he just drank. After a week, Barry Marshall's mom, he asked to the Barry, "Hey Barry, I smell very bad. I can sense very bad breath from you. Just go and check." Then he got that, okay, this should be because of the bacteria. Then he went to the endoscopy. He developed ulcers. What he got? He got the antibiotics. He cured it, and then he isolated the bacteria from his sample, and then he did the samples, and then he intake antibiotics. Believe it or not, in 1990 he got the Nobel Prize for this bacteria. From there, this bacteria comes in the limelight. So this is the very interesting. This is the hero introduction. You see the very beautiful nature of this H. pylori, transmission electron microscopy. Image of Helicobacter pylori, gastric pathogen, as you have 5 to 6 flagellus in the name in the bacteria. So this is how the bacteria, the story, started from. It is the unusual thought. Unusual factor. We don't know this thought. At least 50% of the population, they harbors the bacteria in the stomach. The thing is, if you have the gastritis, if you have the ulcer, then the problem starts. And then the researchers, they believe the Helicobacter is also one of the part of the human gut microbiome. So if you have any other condition or complications, there the problem emerges. So it is associated with the gastritis and ulcer. However, a prolonged gastritis. So that's why people used to say that if you have a condition, prolonged condition like a burp or a GERD, that what we call reflux disease, and then a gastric acid reflux disease, or whatever the condition in your stomach, please go and find a cure, because a prolonged harboring of this pathogen will result in intestinal metaplasia that results in gastric cancer. That's why the World Health Organization classified H. pylori as a class 1 carcinogen. Very few bacteria which is classified as a carcinogen, and H. pylori is one of them. So be careful with that. And as we can see here, a funny cartoon, the H. pylori, it is species on the human stomach, that is ammonia. The ammonia is the one of the very dangerous substance that induce the the lesions. Because of the urease, the enzyme that causes the gastric layer, and that helps the bacteria to survive in the acidic environment. As you know that ammonia, which produces the alkaline pH, that turn the acidic pH into little alkaline, that favors the bacterial growth. So the drug, what we have, like omeprazole, which is the PPA, and then the drug clarithromycin, amoxicillin. Clarithromycin is the macrolide antibiotic, which is a modified version of a macrolide antibiotic to make it survive in the acidic environment. And then amoxicillin, one of the beta-lactam antibiotic. So this is the what we call triple therapy for the H. pylori. However, according to the World Health Organization, what we can see here, the clarithromycin resistant H. pylori is just emerging. So that's how the work starts. Based on, okay, this is a background of our story, and then let's get into the topic. Repurposing niclosamide to combat H. pylori. So the niclosamide, one of the, as we discussed previously, it is one of the drug that they prescribing for the tapeworm infections, and then we got this drug as a MRSA hit, and then I told you, like, I found this drug as also inhibit the H. pylori, and then where it sees the MIC level is really low than the other antibiotics. Other drugs what we have tested. However, still antibiotics like amoxicillin and clarithromycin, they are really good. However, since due to the resistance mechanism, the clarithromycin, so we are seeing this, the drug can be a potential addition. And I did this, based on the MICs, as you all know that we serially dilute the compounds, and then adding the bacteria on the compounds, and then the concentration where it inhibits the bacteria, what we call as minimum inhibitory concentration. As you can see here, the concentration at 64, that you can see here there is no bacterial growth, what we call as minimum inhibitory concentration. So that's how the drug come to our study, our attention. We can't proceed further, and then the bacteria, which is the bacteriostatic in nature, in high density, when there is a high density, like I tested in 10 to the power of 8 cells, number of cells. However, it reduced the CFU of a 1 CFU level, at least like a 0.50 CFU. And how we detect that? So when there is a concentration increase, there will be a lesser percentage of cells, and that's how we decide what is the concentration that the antibiotic that can able to stop the bacterial growth or to kill the bacterial cells. And that's how we say. And then we found this drug. It can inhibit the bacterial growth, and the next study. So yes, it inhibits the bacteria, and then, however, what is the acid stability? So as you can imagine, the H. pylori is a stomach bacteria, gastric bacteria, and then if a drug can go inside and it should survive and stable, it should stable at that concentration, that pH level, acidic pH level. So we tested until 5.5, that is the least minimum acidic condition where the bacteria can grow in vitro, that means in test tube. So And as you can see here, the clarithromycin, the fold increase, I mean the MIC is little, I mean up to 4-fold have increased in clarithromycin and 2-fold increase in amoxicillin. However, as you can see, this drug, which is acid stable drug, it can be stable, and then it can be good if you are prescribing to the condition, to the conditions. And then the next level, the next study, if it is stable in acidic condition, and what should be the resistant level, whether the drug emerge resistance against the drug. So how we have to detect? So in order to detect that, in order to prove the fact whether the drug is not resistant against whether the bacteria is resistant against the cannot emerge resistant against the drug, we culture the bacteria in the presence of drug for 30 continuous days without any break. 30 continuous days, continuous supply of the bacteria. That's how the antibiotic resistance develop. However, as you can see, there is no even a single fold of MIC have developed. So that's how we can say that this drug can be a good candidate to develop as an anti-H. pylori medication. And then the motility inhibition. So in order to colonize, the motility is the very important factors. As you can see here, we have 4 locomatory flagella present in the one side of the H. pylori that induce the spiral movement. The spiral movement can penetrate and kind of drill mechanism. As you can see here, that drill will results in the getting entry into the gastric mucosa layer that results in attached to the epithelial cells. So we found that up to very less 200 micrograms, which is the less than the MIC, it can, even if it is sub MIC, it can inhibit the bacterial virulence factors. That's how we can see here. This is the bacterial slab, the bacterial agar slab. As you can see, there is a lot of bacterial growth over there. However, as you can see here, in the 200 micrograms, there will be a less growth. The swarming movement because of this flagella movement. And you can refer here easily how the bacteria can swim across the medium. Our drug at sub MIC level, it inhibits the motility. That means that there is a less chance of infection induction. And then adhesion and invasion. So the rule of thumb is adhesion is the initial factor of bacterial colonization. So we need to prove whether the adhesion was disturbed. So what we did, we got the gastric cell lines, and then we added the bacteria to the cell lines, and then after some time, we let the bacteria to adhere on the mammalian cells, and after adhesion, we administer the drug. As you can see here at the niclosamide, you can see 1, 2, 3, 4 CFUs. CFU means 4 log 10 CFU. That means for 40,000 cells 40 lakhs of cells that can be washed off or killed from the already adhered cells, epithelial cells. As you can see here, this is the adhered mechanism and invasion mechanism. This is experimentally illustrated that how we did. So this is the cell. We add the bacteria, and then after giving it 2 hours, we wash off the cells that to remove the extracellular cells, and then add gentamicin, because the adhered bacteria during the infection can be killed by the gentamicin, one of the bactericidal antibiotic, and then the invaded bacteria can go into the. So after invasion, we add the drug. So the drug can go inside the epithelial cells and then can decrease can kill the intracellular bacteria. That's what we found, that it should be a good antibiotic. It should be a good candidate. It inhibits. It eradicates the adhered bacteria, and then it kills the intracellular bacteria. Invaded bacteria. So probably a good candidate. And then after the thing, we need to see whether how this antibiotic works structurally and then phenotypically. So during the phenotypes, as we can see, this is no treatment. As we can see here, the bacteria, and then you can see the flagella. When there is a 4x of MIC administered, the bacterial cells, it changes its morphology. That means that it cannot able to retain its structure. That results in it cannot be no longer infective. So like that, at 4x and 1x concentration, as you can see here, and the IJ and KL panels, they are control antibiotics. This is the amoxicillin, and this is CCP. One of the other condition what we need to see the control level. So it is clearly explained in the images that drug works whatever, but we need to find the mechanism of action. However, before going to the mechanism of action, we needed to see whether it is working or not, and then it is fantastically working, and it is proven microscopically. After we did the scanning electron microscopy, making us more exciting about the project, and then doing a lot of work. And then, as I told you before about the infection assay, we added the cells. What we called monolayer. The gastric epithelial cell was plated, and then after 24 hours, we added the bacteria, and then we saw the vacuolization. So simply, as we are seeing here, mammalian cells infected with the bacteria, but regardless, irrelevant. But this is relevant. But I just want to visualize how it works, how it seems. So as you can see here, this is a mammalian cell, and then when the mammalian cell, the size comparison between the mammalian cells and the bacteria. So based on the bacterial adhesion. After adhesion, the bacteria which produce a lot of toxins. So one of the main two toxins are called CAG, which is a cytotoxin associated antigen. Cytotoxin antigen, that is one of the oncogenic protein. As I told you before, the prolonged harboring of this pathogen will result in gastric cancer. So this cytotoxin associated protein, that antigen, which is the oncogenic protein, continuous injection of this protein getting into the cells, which results in modification of signal transduction mechanism inside our gastric epithelial cell, results in metaplasia and dysplasia, and results in gastric cancer. And one other toxin, which is known as vacuolation toxin. As we can see here, this is the normal cell lines, normal gastric epithelial cell lines, and it is nicely formed, a good shaped or uniform shape. And then, as you can see here, all the infected cells after addition of bacteria, see the small tiny vacuoles present in the cell. That is because of the culprit vacuolating toxin, which is called VAC-A. That vacuolating toxin, which is secreted in the surrounding medium, that get into the cells, which results that will act on the mitochondria of the cells, and then plays with the signal transduction mechanism inside the cells, and results in cell apoptosis. And then this is the untreated cells, and after treatment, so the cells infected with bacteria, and after infection, we wash off the cells and then added niclosamide. As you can see here, there is no vacuolation, not even a single vacuolating present inside the cell that was rescued. That's all the cells are rescued by the niclosamide. So that results in that may giving the clue that why it can be a good candidate, because of this all the experiments what we have did. And then, yes, based on the upper panel, that inhibiting the vacuolating toxin. How? Whether it inhibit the toxin entry, or it inhibit the toxin secretion. So we did the transcription translation studies, as you can see here, based on the the densitometric assays at 200 micrograms of 200 nanograms of a drug, it inhibiting the vaccine toxylation translation thing. So after mRNA, after DNA encoding to mRNA, so it stops the mRNA translation, that results in less protein translation. So this is not, the drug. So we have the other clue that the drug can working with or can acting along with the bacterial mechanism and the inhibiting mechanism, antivirulence mechanism, or inhibiting the toxin secretion. So that's how we found this. This can be an anti-toxin molecule. It inhibits the toxin, as you can see here. And then, so next thing, we need to find the mechanism of action. That is the rule of thumb. Whatever the antibiotic you do, whatever the drug you find, we need to find the mechanism of action. Um, the drug, based on the previous experiments, the drug, it didn't inhibit the, it didn't kill the bacteria. It did not damage the bacterial, as you can see here, it did not damage the bacterial cell wall. And I don't know whether it is inhibiting the bacterial DNA mechanism or something, a mechanism inhibiting the DNA mechanism or RNA mechanism. However, I came up with a different solution, that why this drug can act to play with the bacterial membrane, bacterial transmembrane proton motive force, which is the main force that can act with the one of the mechanism that keep the bacteria still running. Based on the mechanism of action, we have, a lot of antibiotics, they have they inhibiting the cell wall of the bacteria. So let's say for an example, beta-lactam antibiotics, simply first antibiotic what we have ever called the penicillin, one of the cell wall synthesizing inhibiting antibiotic, and then vancomycin cephalosporins, they inhibiting the cell wall. However, we have the other group which is called the tetracycline and the macrolides. Those inhibiting the protein synthesis, they working with the 30th subunit of ribosome, that inhibiting that ribosomes, and that results in the bacterial protein inhibition, translation inhibition. And we have one other antibiotic, that is the DNA or RNA synthesis inhibitory antibiotics, like quinones that inhibit, like say for an example, ciprofloxacin, that inhibit the DNA gyrase activity. And then we have one more antibiotic, like repurposing, which is a well-known antibiotic for Mycobacterium tuberculosis, tuberculosis inhibiting antibiotic, which results in mRNA synthesis inhibition. And then we have one more stuff, what we call the polymyxin, that damage the bacterial cell membrane. So as you know that, as you just imagine the fluid mosaic model of plasma membrane, which is the intact membrane, it doesn't allow anything going inside. The antibiotic polymyxin, it targets the membrane, it damages the membrane, cause physical injury, that results in the materials present in the cell, inside the cell, that will ooze out, results in cell death. So apart from all other antibiotic, we have one more thing that we call proton motive force. It inhibits the proton motive force. How we can't see here. So there is a very specialized probe, a fluorescent probe, BCF. That probe, the DAC membrane potential probe, that results in it decrease the membrane potential, results in cell death. The cell metabolic rate was decreased, and then there were no more infection. So that's how we found that the bacteria, the antibiotic that was that can work against bacteria by disturbing the proton motive force. Then the cell toxicity. So, and all you pharmacy students, I don't need to tell more about this kind of absorption, the distribution, metabolism, excretion thing. So that is the other important factor. Whatever the drug you're developing, this is the most important thing. This niclosamide, it seems to be little toxic. However, the antibiotic which is not toxic to the human RBCs because of poor absorption, and the drug is well known that the drug is a poorly absorbed antibiotic that results in, if you are prescribing the drug, that will act locally on the surface of the bacterial, the human host epithelial cell, and that results in bacterial eradication. That's what we hope for. However, there is a studies that making the drug to more absorbable to make it available to the human system, to the plasma level, that will result in even enhanced tissue penetration, that results in good blood availability level. So still the results, the studies are going on. And here I really want to present one interesting model, which is called wax moth as an infection model. So we you know what we did in vitro. We proved the drug is antibiotic, and then we used human gastric epithelial cells to prove the drug can eradicate or can clear the infection from the adhesion, and always on also an intracellular level. And then we proved the proved mechanism of action. The next level is we need to see whether the drug, the efficacy, we need to see the efficacy, whether the drug can rescue the infection model from the infection. So the fly, as you, and in the upper panel, you can see here this called Galeria melonella, one of the wax moth model. So we use that larva as an infection model. It is very simple model and very easy model, and then very cost effective, very cheap model. All we need is the drug, the bacteria, and the larvae. As you can see here, that in the D panel, a person is injecting, so it's like same injection. So everyone have this the excitement about the injection, and we need to do the injection. So before going for the mice level, we can use this as an potential infection model, and this is accepting as an in vivo model. So what we did, we took the Galeria, and then we inject the bacteria in the one left last row, like, and then we infect the, we inject the drug at 25 micrograms, 25 milligrams per kg. So we calculated that level, and then we inject the drug. And then yes, as you can see here, up to 70% of infected larva was rescued. That means 70% survival. 70% survival was recorded. That means, why not? It can be a good candidate. So that's how we finished this study with the infection model. And then, as you may also utilize this model, it's very cheap, and you can use it in your research. And then let's come to the summary. Based on the drug, which is a low toxicity, as I said previously, because of the poor absorption, however, research is going on to make it a good absorbed antibiotic and drug. And based on the results we after published, we filed a patent, and it's still in progress, to use a niclosamide as a drug candidate against H. pylori in human. And we need to initiate the clinical trials also. So it's all in a paperwork. Finally, repurposing, it's, as I said previously, it's time, cost, and the effort saving. You can save a lot of efforts, and still there is a lot of rooms to play. So we have you can see that whether it may be whatever it may be, HIV drug, can be anti-cancer drug, and I forgot to say this niclosamide is one of the anti-cancer drug also. I mean, this drug, the research are ongoing, and then people have filed in the clinical trials. They are using niclosamide as an anti-cancer drug. So still, I said there is a lot of room to play. It's up to you whether you can play or ignore about the work. So I sincerely want to thank, none other my dad and my amma papa. So all my gratitude and my very happy moments always sharing with my father and mother, my sister, and my wife and my baby. And I must thank Dr. Lefterius Mylonakis. He's my mentor right now, and my team, Beth Fuchs and Gerald Now and Louis Rice, they are in Rhode Hospital, and I really want to. I don't want to thank. I need the blessings. So Mardamuthu Sundaram, he's the retired principal scientist in Chikri. So I just spent 4 months with him. You can't even imagine, even a single minute can change your life. So my life has been changed by Mardamuthu Sundaram sir. He is a very fantastic scientist. He simply says that உனக்கு வேலையை மட்டும் பாரு. உனக்கு வேலையாகுனா நீ ஒர்க்கை மட்டும், உனக்கு ரிசர்ச் வேலையாகணும்னா என்ன வேணா பண்ணு. ஒர்க்கை பொறுத்தவரை. That means that level of dedications he seeded in me. And also my guru, Kim Jong-bae, professor in the Yonsei University, South Korea. So he's my PhD thesis advisor. Because of him, only because of him, I successfully completed my PhD, and I'm here because of him. And I also extend my thanks to Department of Microbiology, Providence University, and Department of Biomedical Laboratory Science in Yonsei University, and friends, families, colleagues, and collaborators. And I thank everyone. And thank you for this session. And if you have any questions, please shoot out. Thank you very much.
Thank you, sir.
Yeah, welcome.
Any questions? So good morning, sir.
Yep.
Sir, the topic is about the repurposing of drugs against infectious diseases.
Yes.
So, what is the need of repurposing for infectious diseases, sir?
See, the main theme of this work is antibiotic resistance. As I said previously, the emerging antibiotic resistance, which is the high demand that highly demands the new drug. However, the bacteria is really they are really brilliant than us. We all emerge from single cellular to multicellular to the multi-organ beings. However, the bacteria, they are really brilliant. They know how to deal with the problem, and because of that, I will tell you a simple story that methicillin, which is one of the first synthesized antibiotic, within 1-2 years of development, the bacteria Staphylococcus aureus it developed the resistance against the drug. Still, MRSA is the highest number of causing highest number of fatalities. So because of this antibiotic resistance, we need a new drug. However, if we go for a new drug development, it need 20 years, at least 15 years, and then it costs a lot of money. However, drug repurposing will cut short all other expenses, time, everything, etc. So that's why I suggest go for a drug repurposing, and then that it's really working. And I'm seeing this, if you say for vancomycin is the gold standard antibiotic, which is a last resort antibiotic of against Staphylococcus aureus. As you can see, the 1 or 2 is the MIC, 1 and 2 micrograms per ml is the MIC. However, the niclosamide or ornithine, which produce like 0.25, which is 8-fold lesser than the clinical antibiotic. So we don't know the potential. If we still explore, we can find a lot of potential of this drug, repurposing drug, we can cut short the effort and we can reduce the time. It can come easily toward the market. So that's why I prefer drug repurposing for the infectious disease. And that is also helpful. As you can see here, a very best example, remdesivir. So remdesivir, which is one of the GILEAD pharma company's drug, initially they developed for the Ebola, and now they are using for SARS-CoV. Within a very short duration, they found that repurposing, the purpose of the potential of repurpose, and they are using. Believe it or not, in US, United States, they are prescribing for very critically ill patients. So that's why I choose repurposing. I suggest.
Thank you, sir. So if you see the drug discovery in the field of antibiotics, so last 10 to 15 years, there is no major commercial antibiotics, sir.
Exactly. That's also.
What is the reason behind this, sir?
So I'm saying that still every antibiotic that need at least 15 years of time consuming that need a lot of time, and then the FDA they establish a policy that we need to cross like phase 1, phase 2, phase 3, and phase 4. These phase 1, phase 2, that is fine, but phase 3, which we need to recruit a lot number of, at least thousands of patients, and phase 4, which having thousands of patients, that is always a time consuming. And also people, they are thinking that, so to get a potential antibiotic, it should be a resistance. It should not be develop any resistance. So the last antibiotic, what I can say, that linezolid, that is one of the oxylidone antibiotic. That is the last commercial antibiotic that was available for right now. Since because of these lot of limitations, we are facing this problem, and also most of the antibiotics, they are from natural resources, like most of the actinomycetes. Until now we have a lot of antibiotics they are producing from actinomycetes. People still they're probably they may be fighting against the actinomycetes. That may be the reason they don't get any antibiotic. However, still lot of drugs they are in the phase 2 or 3 or 4 trials. Because of the policies, we need a lot of time to develop. That's the reason I'm seeing here.
Thank you, sir. Thank you, sir.
Yep.
Sir, I have told antibiotic resistance or sensitivity. So last few years we are seeing a lot of superbugs which cannot be easily treated by antibiotic.
Yep.
So how to overcome these things? Because that the pharmacists are going to play the major role in antibiotic stewardship.
Let me tell you one story. 2017 or 2018, there was a woman in United States, probably she's from Philadelphia or Kentucky. I'm not sure about the state. She was infected with the Klebsiella pneumoniae. The bacteria, which is what she got, is a Klebsiella pneumoniae, and then she was admitted in the hospital. The hospital staff, they isolate the bacteria and then they run antibiotic sensitivity. Believe it or not, there is a 26 approved antibiotics in this country. That drug resistant to all 26 antibiotics. Unfortunately, that patient ended up, she is no more right now. That's what I'm saying right now. This is an emerging problem. This is an emerging problem, very emerging problem. We should take care of this. You know, the cancer death, the antibiotic resistant death will move forward, and it can exceed the cancer death in 2050. That's what the 2050. That's what the estimates are saying. So the main reason I'm telling you here to all fellow students, all my fellow students and colleagues, please ever don't go to the pharmacy and ask antibiotics from pharmacist, because overuse of antibiotics and uncontinued antibiotic usage and unprescribed use of antibiotic, that is the main reason of antibiotic resistance. If the doctor prescribes 5 days of course, everyone should continue this 5 days of drug. If someone skips the drug within 2 or 3 days after they feel, oh, I'm good right now, please don't do that. Second thing, if you are a potential pharmacist, in few years, please don't ever give any antibiotics without prescription. We need to save the antibiotics. You know, can you believe this? There is a lot of groups, and then the agencies, the non-profit organizations, they say, they develop a motto of saving antibiotics. We need to save the antibiotics. So please ever don't give any antibiotics without prescription, and don't ever get the antibiotics in the pharmacy. And then please use. Please finish the course if you are taking any antibiotics, and then suggest all these things to your colleagues and friends. So that's how we should save the antibiotics. We have very limited antibiotics right now. We need to save that. So this is all my thoughts about the question what you said.
Thank you, sir. So if you see last few infectious diseases like dengue, H1N1, like during the cases we don't have a treatment of initially, but that our natural remedies, which has been played a major role in controlling those diseases. So any possibility of repurposing those things, or what is the role of natural remedies?
So let me tell you this. So H1N1, dengue, so those comes under viruses, and then that H1N1, we have potential flu vaccine here. However, due to the antigenic shift, every year, I mean, the season, we have the mutated virus, and the pharmacy companies, they're working on producing the the vaccines every year with the new strains that will save you from the infection. So vaccine is the first thing. Second thing, dengue vaccine. Dengue vaccine, still now people, they don't have succeeded on the dengue vaccines. However, as you said, natural remedies. Actually, my PhD topic, what I did PhD, to see the effect of piperine against H. pylori. So piperine is the major constituent in black pepper. We have a statement in our place. I mean, in Tamil Nadu, பத்து மிளகு இருந்தால் பகைவன் வீட்டிலும் சாப்பிடலாம். This is kind of a statement in our place. It's a saying, or it's a word from mouth. So the pepper, which is having the very enormous thing to rescue from the infection. So that's what in my mind when I was starting my PhD in 2012. I took that piperine, and then I did assays, and I found that you know what, that's the first report. That's the first ever report, in the world that piperine inhibits the H. pylori, which is the gastric pathogen. That's the first report I published it. And then it is also anti-inflammatory thing, and it is also an anti-cancer thing. So that's what I published. So in answer to the exact thing, we all need researchers to come forward to explore our Siddha medicine, to say, so this content having this single molecule, which is having the enormous value, or very fantastic and exciting and terrific effects against this pathogen. So all we need is, so in Tamil Nadu or India, people, they are doing crude extracts. They all in the form of kashayam, all in the form of suranam or legium. This is all combination of things. But what I prefer, I suggest, what I did, a single compound. So that need a lot of hard work to find, to explore what all the compounds present in a single particular thing, and then we need to explore even as one by one, and a single compound, single molecule, and single compound. Then we need to see. Obviously, I can say that we have our Siddha medicine. We have a lot of Siddha medicines, all in the form of crude extract or combination. We need to explore in a single value. That's how it will be go in a big shot. That's my thought.
Thank you, sir. So you are working in US environment, which is highly regulated market. But like India, so here we are overusing the antibiotics. It's not, it may not be a problem of patients. Even doctors also, if we are going for doctors, they are prescribing the high-end antibiotics initially. So what is your opinion on that, sir?
No, I didn't get your point. I'm sorry.
No, sir. If you are going for a respiratory infection, instead of amoxicillin, they will first start itself, they will go for Fixim or high-end antibiotics.
Exactly.
So what is your opinion on that? Yeah, what is your opinion on that?
See, let me tell this story in my personal case. My daughter, my 3-year-old daughter, she got a cough. I went to a doctor, and then I asked, as I'm from a medical profession, a paramedical stuff, I know, I suspect what is the onset. I mean, based on the onset, I can say that, okay, this is what the infection. I told her, based on the cough, she maybe have like a bacterial infection, or bacterial cough. So please give her antibiotic. And she said, no, we don't know whether it may be viral thing, or it may be viral infection, or maybe bacterial infection. We don't know. We can't sure. So we can't say we can give you any antibiotic. Believe it or not, my daughter ended up 5 days of fever. They didn't give any antibiotic for 5 days. I was giving Tylenol, like a paracetamol and ibuprofen, to reduce the symptoms. However, they just want to monitor what is the exact thing. So once they confirm, wow, this is what going on, yeah, because of, and then after the examination, so they just said, once the fever is for 4 after 4 days, the fever is still persisting, then we give the antibiotic. So after 4 days, they examined, and then they said that, okay, this is the infection, and then they gave her amoxicillin. That means, based on the first inspection, based on the first study, nowadays here in India, the doctors, They are prescribing simply antibiotic and then paracetamol and this, and then yeah, just go for 5 days. That's it. That is the main reason of antibiotic resistance. So based on the practice here in this country, what I'm seeing here, they don't give you antibiotic at the very first moment. They just want to monitor you, and then they will give you. Even if you say that, oh, I'm very much illness, I'm very much facing this kind of thing, what I am facing fatality, don't worry. Even if you face that much of thing, they have a lot of facilities to rescue you. However, the thing is, based on the first inspection, they don't give you antibiotic. That is the thing what I'm seeing here.
Thank you, sir. So what is your opinion on usage of antibiotics in poultry, sir? Poultry.
So I'm sorry to say this, poultry and usage of antibiotics in animal industry, that is the main one of the main reason of antibiotic resistance. And there is one study that if you are prescribing antibiotic, the antibiotic excrete, the antibiotic residues will go to the residues and will go to the excretion, and then that will result in ended up in the soil pathogen. They develop resistance, and based on the transfer, plasmid transfer mechanism or horizontal gene transfer, the antibiotic resistance will transfer from one bacteria to another bacteria. So in order to prevent this, what is my suggestion is, in the poultry, we should segregate. We should divide two categories. One is antibiotic should only use to human, and antibiotic group that should only use to the poultry or animal industry. The thing is, I'm seeing people in the animal industry, they're using tetracycline. Tetracycline is one of the class of antibiotic that inhibit the bacterial ribosomal translation mechanism. It inhibits that. So if you develop resistance against tetracycline, even if you give doxycycline, it won't work, because both of them comes in the same class. Likewise, colistin is one of the antibiotic, which is one of the forgotten antibiotic they're using in poultry. So if you develop anything like colistin, that is called MCR, so the MCR gene which is responsible for the colistin resistance. However, this thing can be interfere with the other antibiotic groups. So what I'm seeing here, what I'm suggesting, so if you are using the antibiotic to the animal, that should be in a separate class. That should not be with the human antibiotic consumption class. So if you follow this, we can prevent the antibiotic resistance emergence from animal to human.
Thank you, sir. So if we see that current pandemic situation, if we see the current pandemic situation, sir, there are a lot of repurposing drugs has been used for the treatment.
Yep.
So in the case, the repurposing drugs has to be clinical trial. We should done clinical trial and we have to use, or we can use depending upon the emergency, sir.
We need, that's what the problem is. Even for this kind of pandemic, this need to be done. So probably they can do some fast track approval or fast track applications. However, the FDA, they strictly mention that they should go for this level. Though that's how the remdesivir. So they are not giving actively, and then the remdesivir, there are people in that condition, so the people they are using that, but it is not using as much as possible, because viral disease is something, it's so different than the bacterial disease. That's how we use vaccines. So in order to pursue with the vaccines, we need to see lots and lots of vectors. So the vaccines, they deal with the immune mechanism. So that is very complicated mechanism. That the delivery drug, the delivery vehicle. The delivery vehicle may play a very important role. So that's how the Moderna is one of the company, they are producing the SARS-CoV vaccine. They are in clinical trial stage 4. They presumably, they're thinking that okay, they can survive or they can succeed with that. So hopefully we are expecting that. However, even though in this condition repurposing, they need something kind of fast track approval or fast track application that should be submitted, and then it can go through something like that. That's what I'm saying.
Thank you, sir. Students, any other question?
Thank you, sir. Thank you, Dr. Nagendran, sir.
Yes, sir.
Really, we had a